Hello everyone, my name is Matthew Jones and I'm a master's student at Western University. Today I'm going to be presenting my research project regarding how the endocannabinoid system plays a role in anxiety. In my project, this is done through modulating levels of the endocannabinoid ligand anandamide using a protein carrier inhibitor for fatty acid binding protein 5 or FABP5. In doing so, it allows the study of how the endocannabinoid system modulates amygdala activity and how this impacts connected limbic regions such as the prefrontal cortex and hippocampus to regulate anxiety-related behavior and neuronal activity states. So today I would like to highlight 3 learning objectives. The first concerns a basic introduction to the endocannabinoid system, followed by how the brain area of focus, called the basolateral amygdala or BLA, is involved in anxiety-related processing. Finally, we will branch these two concepts together to explore how pharmacologically increasing levels of the endocannabinoid ligand anandamide through inhibition of its breakdown might offer a new therapeutic approach to treating anxiety. So as mentioned, the endocannabinoid system is the focus of this research, or a focus of this research project, and it's composed of endocannabinoid ligands such as anandamide or AEA, which are endogenous lipid-based retrograde neurotransmitters that bind cannabinoid receptors. The endocannabinoid system is a huge emotional regulator within the brain, importantly for my research playing a large role in anxiety regulation, stress coping, and fear reactions. As mentioned, anandamide is the key ligand in this system, and it's been shown to act in a retrograde manner to suppress glutamatergic release, among other synaptic mechanisms. This has a functional role in regulating brain circuits involved in anxiety processing, including those connected with the BLA. Now within the BLA, glutamatergic signaling regulates anxiety behaviors through modulation of pyramidal neurons. These pyramidal neurons are part of a bidirectional circuit with the PFC, where BLA to PFC interconnections are implicated in the regulation of anxiety and stress, and are indeed modulated by the endocannabinoid system. The BLA also forms connections with other limbic areas, such as the hippocampus, to modulate other facets of behavior along with anxiety regulation. So the main focus of this research is then looking into anxiety disorders and potential treatment. So anxiety disorders are defined by excessive worrying, uneasiness, and fear of future events to the point where it affects social and occupational functioning. These are known to impact nearly 1 in 3 people, so they are fairly prevalent. Presently, one of the most common drug groups prescribed for anxiety disorders are benzodiazepines due to their short onset of action and efficacy. However, due to the fact that they rely on widespread GABA modulation, they tend to cause many unwanted side effects, as you see in the figure on the right. So we're looking into alternate options, given this is not a great treatment choice. Interestingly, dysfunction of the endocannabinoid system is involved in many psychiatric disorders. Given it's a much more specialized system than GABA, it has a lot of promise for minimal offhand side effects. Now, in the past, neuronal plasticity has been associated, at least in terms of anxiety, with the cannabinoid 1 receptor mechanisms. Tan et al. Found stimulation of the BLA produced plasticity changes along the BLA to PFC pathway, which was dose-dependently blocked using a CB1 receptor antagonist. Now, similar results are lacking with antagonists of other endocannabinoid receptors, such as the cannabinoid 2 receptor, CB2R, and JBR55 receptor systems, although they are very much receptive and responsive to anandamide binding. So I'd like to see how they may play a modulatory role in anxiety processing, given along with the cannabinoid 1 receptors show high expression in the BLA, PFC, and surrounding limbic regions, which have large roles in anxiety and fear processing. So now that we've gone over the receptors anandamide might activate, let's get back to the ligand itself. How do we modulate anandamide? So after exerting action at receptors, signaling is terminated by presynaptic reuptake, by degradation by the enzyme fatty acid amide hydrolase, or FAAH there. Anandamide is a hydrophobic lipid, so its diffusion is limited and transport is facilitated by protein carriers known as the fatty acid binding proteins, FABPs. Now, FABPs do play a crucial role here, and Tan et al. In 2014 demonstrated that inhibition of the FABPs elevates anandamide levels. So there are many different subtypes of FAB-Bs, but here we're going to focus on FAB-B5, given it displays one of the highest affinities for anandamide and is prevalent within BLA and PFC regions. So for this project, I'm using a novel pharmacological inhibitor of FAB-B5, which is SBFI-103, as an exogenous mechanism to control endogenous anandamide levels. So the present goal of my research project is therefore to examine the effects of localized inhibition of FAB-B5 signaling within the BLA on anxiety and fear-related behavior processing. I will also examine how modulation of neuronal activity states in the BLA to PFC and BLA to VHIP ventral hippocampus pathways may be involved in anxiety, and the mechanistic biomarkers that are implicated in intra-BLA anandamide signaling. The central hypothesis of this research project, then, is that anandamide decreases glutamatergic activity in the BLA, which is responsible for anxiety-related symptoms. I thus predict that increasing anandamide levels in the BLA through FAB-B5 inhibition will induce an anxiolytic effect, as observed by measured changes in behavioral outcomes and neuronal activity dynamics. So, just quickly on to methods. First, I performed in vivo electrophysiology. So this included microinfusion of the drug into the BLA of rats, and then voltage clamp recording of single neurons in the prefrontal cortex and ventral hippocampus to determine regulatory effects of endocannabinoid transmission on these neuronal pathways. So here I looked at firing frequency, bursting rates, along with local field potential analyses for the oscillatory pattern changes following FAB-B5 inhibition and antagonist administration. So next I'll be performing behavioral analyses, which include surgically implanted guide cannulae to locally administer the FAB-B5 inhibitor to the BLA. I'm going to look at anxiolytic effects using established behavioral assays, and then the effects of the FAB-B5 inhibitor will be examined in 4 different treatment groups, the same as the electrophysiology. So that's the FAB-B5 inhibitor alone, and then co-administered with one of the 3 receptor antagonists. I'll also have a vehicle control group, which will receive a vehicle infusion. So now on to discussion of the results I've gathered thus far. So as I mentioned, I'm looking into the effects of local FAB-B5 inhibition in the BLA on, which we see on the left here, on the firing dynamics of the prefrontal cortex, the purple box on the left, and the ventral hippocampus, the purple box on the right. So following 5 to 10 minutes of baseline recording, I infused 0.5 microliters of drug into the BLA and then recorded for another 30 minutes to determine the effects that were observed. So first, we'll take a look at the prefrontal cortex results. So starting off, we can see quite clearly that following infusion of the FAB-B5 inhibitor, a large majority of neurons in the prefrontal cortex respond with an increase in firing rate. This contrasts really well with the vehicle control pie chart, which we see on the right, where very few neurons actually displayed an increase following the infusion of the drug to the BLA. The third pie chart on the bottom there shows the distribution of firing rate effects when the FAB-B5 inhibitor was administered with an anandamide synthesis inhibitor. In other words, lower levels of anandamide were present. So this kind of shows that the anandamide was responsible for the observed effects that we see here, because we are unable to see the same consistent increase in firing rate when anandamide synthesis is inhibited. So then if we look at the receptor antagonist groups, we can see that in the cannabinoid 2 receptor group, the effect certainly looks blocked. There also seems to be a mild block in the cannabinoid 1 receptor group, but then if we look at the GPR55 antagonist, it doesn't look as though it's producing a very strong block, an effect of the FAB-B5 inhibitor. So putting this into numbers, we can see the changes more clearly. So we see the FAB-B5 inhibitor produces a clear increase in the firing rate of prefrontal cortex neurons. This effect is not blocked by the GPR55 antagonists, so we can assume that this receptor isn't involved in the effect. The CB1 receptor antagonist appears no different from vehicle control, along with the CB2 receptor antagonist, which is producing a complete block. So from this data, it appears as though the cannabinoid 2 receptor is principally involved in a BLA to PFC pathway regulation, with certain neuronal populations also depending on cannabinoid 1 receptor modulation. So this data shows the timeline of effect, with the FABB5 inhibitor being compared to both the vehicle control group and an anandamide synthesis inhibitor control group. So the effect seems to be most prominent within 10 to 20 minutes following BLA microinfusion. So finally, for prefrontal cortex region, we're going to look at the local field potential recordings of oscillatory pattern changes. So whereas previously we were looking at spike frequency of single neurons, local field potential analyses, we're looking at the changes in extracellular electric potential to determine synchronous wave activity patterns in that area. So specifically, we're looking at high gamma wave activity, which is associated with cognitive phenomena such as working memory, attention, and perceptual grouping. Now, interestingly, a past study showed altered gamma wave activity in mood disorders during emotional behavior tasks, so it's interesting that the novel drug is producing an effect here. And what's most interesting, actually, is that the effect is only seen when the cannabinoid 1 receptor is blocked, which may point to a role of the cannabinoid 1 receptor in reducing or regulating gamma wave activity. Now, moving on to the ventral hippocampus, we can see that there are some differences. So the effect is definitely more split, although we can see that there is a clear difference from the vehicle control and CB2 receptor antagonist groups. As for the CB1 receptor and GPR55 antagonists, most neurons saw a change in firing frequency, with the majority actually increasing following BLA microinfusion. This is what it looks like in numbers, and so we actually see significant increases in both CB1 receptor and GPR55 receptor antagonist groups. So this indicates to me that they might be inhibitory and therefore responsible for decreasing hippocampal firing rate, as when they're blocked, we don't see the same split that is observed with the FABB5 inhibitor alone. So from these results, I have two main takeaways. So first, when microfused into the BLA, the drug is producing an effect in PFC and ventral hippocampus regions, seen through changes in firing frequency and oscillatory patterns. This is impacted by the co-administration of receptor antagonists, which gives strong indication for the accountability of the CB2 receptor in this effect. Now, this is a novel result, as research regarding the impact of the CB2 receptor on behavior is quite limited. Ongoing research in my lab regarding the behavioral effects of FABB5 infusion to the prefrontal cortex also indicate responsibility of the CB2 receptor. So this is something that is of note given the highly restricted expression of the CB2 receptor to specific brain areas that may allow for modulation of the receptor whilst avoiding many side effects produced with typical medications. Therefore, this receptor will be a clear focus of study going forward. Second, looking at each 5-minute segment separately is necessary to gauge a timeline of effect for this drug. And this is mainly used for when I begin behavioral testing, so I know when to begin the behavioral test and that the drug has taken full effect. So having seen these effects, the next step will be to collect behavioral data regarding the effects of FABB5 inhibition in the BLA. Furthermore, I'll be performing molecular analyses in the form of Western blotting to investigate downstream protein phosphorylation differences in the BLA following FABB5 inhibition. So overall, with this research, I'm using a novel drug, which is SPFI-103, a FABB5 inhibitor, to advance understanding of how a specific FABB5-mediated transport mechanism within the brain's anxiety processing might modulate anxiety-related behaviors, neuronal circuits, and specific molecular biomarkers. These studies are examining the neurobiological mechanisms regulating anxiety and how the endocannabinoid system might functionally regulate these neural and psychological phenomena. In conclusion, in using a novel drug to inhibit FABB5 activity, one of the principal protein carriers for anandamide degradation in the BLA, it allows me to study how endocannabinoid modulation impacts anxiety processing and behavior. Thus far, I have found effects at the single neuron and multi-neuron level following infusion of the FABB5 inhibitor. These effects seem to be heavily dependent on the cannabinoid 2 receptor system, which warrants further investigation as this system is largely unexplored thus far. I would like to thank my lab and funding sources, and will now open up the room for questions. Thank you for listening.
Hello everyone, my name is Taigun and I'm a postdoctoral fellow working at the research group of Professor Stephen Laviolette. Today I will be presenting you my research project entitled Identification of a Novel Mechanism Regulating Anxiety Behaviors in the Prefrontal Cortex, Fatty Acid Binding Protein 5 and the CB2 receptor. Endogenous cannabinoid signaling has been linked to modulating fear, anxiety, and stress responses, and its dysfunction is implicated in the pathophysiology of psychiatric disorders, including schizophrenia, substance abuse, depression, and anxiety disorder. Anandamide and 2-AG are endocannabinoid ligands that are synthesized on demand following neuronal activation and are released into the synaptic cleft by diffusion. These ligands activate numerous target receptors, including cannabinoid 1 receptor, cannabinoid 2 receptor, PIR gamma, and GPR55. Thereafter, anandamide and 2-AG are cleared from the synaptic cleft by cellular uptake and metabolized by fatty acid amide hydroxylase and monoacylglycerol lipase, respectively. Importantly, pharmacological agents that increase endocannabinoid signaling, most notably anandamide signaling, have been shown to reduce anxiety, facilitate fear extinction, and dampen amygdala activation in vivo. Pharmacological inhibition of FAAH or anandamide uptake, both of which results in reduced anandamide hydrolysis, thereby increasing synaptic anandamide availability, has been shown to produce anxiolytic-like effects and facilitate fear extinction in rats. Furthermore, a key polymorphism in the FAAH gene, which destabilizes the FAAH protein and increases anandamide signaling, has been associated with reduced trait anxiety and elevated cortex and amygdala connectivity. Altogether, these findings implicate anandamide signaling in anxiety behavior and fear expression. Water-insoluble lipids such as anandamide require chaperone proteins for their intracellular transport. Research has identified FAP-P5 and possibly FAP-P7 as intracellular transporters for anandamide. Intracellularly, FAP-P5 binds to anandamide and transports it to FAAH, which is localized in the endoplasmic reticulum for its breakdown. So similar to the inactivation of FAAH or anandamide uptake, inactivation of FAP-Ps results in elevated anandamide-mediated neurotransmission. Importantly, inhibition of FAP-P5 and FAP-P7 by systemic SPFI26 administration results in reduced ethanol consumption, as well as anti-inflammatory and analgesic effects without causing motor or cognitive impairments in mice. Furthermore, FAP-P5/7 knockout mice demonstrate an antidepressive phenotype. Also, elevated FAP-P5 mRNA levels have been detected in the PFC of postmortem brains of schizophrenia patients. These findings therefore strongly associate FAP-P5 signaling abnormalities with various psychiatric conditions. Current anxiolytic treatments are associated with significant deleterious side effects, including dependence, withdrawal, and cognitive impairments, which are major therapeutic limitations. The endocannabinoid system therefore represents a promising neurobiological target for novel anxiolytic pharmacotherapies. Given that FAP-Ps are key components of the endocannabinoid signaling, we here aim to investigate the possible effects of FAP-P5 inhibition on the modulation of fear and anxiety. If such effects are detected, we also aim to investigate the potential neuronal and molecular mechanisms underlying these effects. Our behavioral procedures start with stereotaxic surgeries. Using male Sprague Dawley rats, we implanted steel guide cannula into the prelimbic region of the medial prefrontal cortex. After recovery, we performed behavioral tests following infusion of a novel compound, SPFI-103. SPFI-103 is a selective inhibitor of FAP-P5 with negligible affinity for FAP-P3 and FAP-P7. Furthermore, we challenged the effects of SPFI-103 with various antagonists of target receptors of anandamide, including CB1 receptor, CB2 receptor, GPR55, and PIR gamma. Each behavioral test started 5 minutes after the microinfusions. Here, using in vivo electrophysiology in anesthetized rats, we examined the neuronal firing and oscillatory patterns in the BLA and ventral hippocampus following SPFI-103 exposure into the prelimbic cortex. Given previous evidence demonstrating that systemic elevation of anandamide induces anxiolytic effects in rodents, we hypothesized that FAP-P5 inhibition within the prelimbic cortex would produce anxiolytic effects through CB1, CB2, GPR55, or PPAR gamma-dependent pathways. Anxiety behaviors were assessed using the elevated plus maze and light-dark box tests. Elevated plus maze is a plus-shaped maze in which two arms are closed and the other two are open. Spending more time in the open arm corresponds to anxiolytic behavior. In the light-dark box, there's a dark and a light chamber, which is illuminated by a lamp. Spending more time in the light chamber corresponds to anxiolytic behavior. As seen in the figures, SPA5103 infusion was dose-dependently anxiolytic as animals receiving 5 microgram per hemisphere. SPA5103 spent more time in the open arm, entered it more frequently, and spent more time in the light chamber and entered it more frequently. Interestingly, SPA5103 induced anxiolytic effects in the EPM and LDB were prevented by co-administration of AM630 and CID 160. These results indicate that intra-PLC FAP-P5 inhibition results in a dose-dependent anxiolytic behavioral phenotype, which is dependent on CB2 and GPR55 mediated mechanisms. Interestingly, CB1 receptor mediated mechanisms do not appear to be involved in these effects. As seen in the top figure, 2 distinct test environments were used for fear acquisition and retrieval. On day 1, animals received 2 acquisition sessions in environment A, during which they received 3 food shocks. On day 2, following microinfusions, animals were placed in a novel environment for 5 minutes to test fear generalization. Immediately afterwards, animals were transferred to the fear environment to test contextual fear memory retrieval. On day 15, animals were placed again in environment A for 5 minutes to test remote fear memory retrieval. 2-way repeated measures ANOVA revealed a significant treatment and environment interaction. Post-hoc analysis showed that SPA5103 infusion reduced contextual fear, and this effect was reversed by co-administration of SR141 and AM630. These findings demonstrate that intra-PLC FAP-P5 inhibition attenuates contextual fear expression through CB1 and CB2 mediated mechanisms. Also, no treatment effect was observed on remote fear recall, suggesting that infusions before the initial fear retention test had no impact on long-term fear memory processing. Pharmacological agents modulating fear, pharmacological agents modulating endocannabinoid signaling may induce changes in locomotion, which could underlie or influence FAP-P5 inhibitor-induced anxiolysis. Using the open field test, our results showed that neither SPA5103 or its co-application with the antagonist affected locomotion or rearing. Furthermore, there was no treatment effect when the antagonists were administered alone, suggesting that these drugs by themselves do not alter anxiety behavior or locomotion. We also investigated the effects of intra-PLC FAP-P5 inhibition on spatial working memory and short-term temporal recognition memory using spontaneous alternation and temporal order object recognition tests. Also, to test whether FAP-P5 inhibition alters reactivity to sensory stimuli, we examined the effects of intra-PLC SPA5103 on prepulse inhibition. Our results show that effective anxiolytic dose of the intra-PLC FAP-P5 inhibitor appears to produce no cognitive performance or sensory-motor gating side effects. The PLC region modulates emotional behavior by exerting top-down control over BLA neuronal activity. Thereafter, long-range BLA projections to PLC and ventral hippocampus control the expression of emotions, most notably fear and anxiety. After isolating pyramidal neurons in the BLA, we analyzed their spiking activity, pre and post intra-PLC infusions, using in vivo electrophysiological recordings. The effects of the infused agents were analyzed as 3 10-minute bins and as 30-minute overall activity. Overall, SPA5103 infusion caused a 31% reduction of overall firing rate, which was prevented by co-administered AM630. Population analysis of PLA neurons demonstrated that 69% of PLA neurons showed decreased activity following SPA5103 infusion, whereas 25% showed decreased activity after vehicle infusion and 17% after the infusion of the combo. These data demonstrate the CB2 receptor-dependent attenuation of PLA neuronal transmission by intra-PLC FAP-B5 inhibition, which possibly underlies FAP-B5 inhibition-induced changes in the expression of fear and anxiety. Given that PLA and ventripocampus inputs modulate anxiety-like behavior, we isolated pyramidal neurons in the CA1 region of the ventripocampus and analyzed their firing frequencies pre and post intra-PLC infusions. Overall, SPA5103 infusion caused a 110% elevation of overall firing rate, which was prevented by co-administered AM630. Population analysis of ventripocampus neurons revealed that 64% of ventripocampus neurons showed elevated activity after SPA5103 infusion, whereas 22% showed elevated activity after vehicle infusion and 27% after the infusion of the combo. These results indicate that intra-PLC FAP-B5 inhibition significantly increases activity of putative ventripocampus pyramidal neurons, which is prevented by CB2 receptor antagonism. Intra-PLC SPA5103 does not modulate mesolimbic dopamine activity states as seen here in the nucleus accumbens shell and ventral tegmental area. So this suggests that FAP-B5 inhibition poses a low risk of addictive liability. In order to investigate the effects of intra-PLC FAP-B5 inhibition on local field potentials in the BLA and ventripocampus, LFPs were recorded concurrently with signal unit activities. In the BLA, SPA5103 induced enhancements were detected in the power of beta and low gamma oscillations, both of which were prevented by AM630 co-administration. In the ventripocampus, SPA5103 induced attenuations were detected in the power of alpha, beta, and low gamma oscillations, all of which were prevented by AM630 co-administration. These oscillatory changes might be involved in the suppression of fear and anxiety-related behaviors. In conclusion, intra-PLC FAP-B5 inhibition reduces anxiety-like behaviors and the expression of contextual fear without affecting locomotor activity, learning, memory performance, or sensory motor gating. We also identified a novel CB2-dependent anxiety processing mechanism directly in the PLC. Intra-PLC FAP-B5 inhibition altered activity of pyramidal neurons in the BLA and VHIB, as well as alpha, beta, and low gamma oscillation patterns within these regions in a CB2 receptor-dependent fashion. So CB2 receptor activation opens postsynaptic calcium-activated chloride channels in rodent medial prefrontal cortex, leading to reduced neuronal excitability and reduced firing frequency in the PFC pyramidal cells. Therefore, mechanistically, FAP-B5 inhibition could activate CB2 receptors in the PFC to reduce excitability of glutamatergic neurons projecting to BLA, dampening its activity. Also, acute FAP-B5 inhibition had no impact on baseline dopamine system activity states, causing less risk for activation of addiction-related brain pathways. So our results provide evidence on safety and tolerability of FAP-B5 inhibition, which may serve as a promising target for the development of novel anxiolytic interventions. Thank you for listening.
Hi, my name is Enzo, and I'm going to present evidence for a synergetic effect of CBD and THC formulation in anxiety and depressive-like symptoms following chronic stress. First, the depressive disorder has been characterized by two main symptoms: the depressive mood and the decrease of pleasure or interest. Also, this disorder has a comorbidity with anxiety disorder, and both pathologies are closely related with stress. preclinical and clinical evidence has shown that chronic stress produces an increase of all of these symptoms. Now, for these pathologies, we have treatment, we have pharmacological treatment. However, this treatment has some limitation. For example, the resistance of the drugs, the side effects, and also the slow onset of the action. For this reason, it's important the discovery for new treatment. For this, preclinical evidence has shown that two main components of the cannabis, the CBD and the THC, shows anxiolytic and antidepressant effect. Interesting, this pharmacological effect has been associated to different molecular targets. For example, for CBD, the anxiolytic and the antidepressant effect has been associated with the serotoninergic agonist, in this case, in the 1-ACE receptor. On the other hand, the THC, the effect of the THC in the mood and the anxiety has been associated with the CB1 agonist. Now, taking together this evidence, it's possible to suggest that the combination of both drugs may produce a synergistic effect in this behavior. For this reason, the aims of my research is to characterize the effect of 101 CBD-THC radio edible formulation in anxiety and depressive-like behavior in a stressed rat. And the second aim is to determine the underlying neurobiological mechanisms. Now, what are the neurobiological mechanisms associated with stress and depression and anxiety? Well, here we can see different brain area that has been associated with these pathologies. And also, evidence has shown that chronic stress produces different changes in this area. For example, for the prefrontal cortex, the chronic stress exposure reduce the synapses in this area and also reduce the neuronal activity. In the case of the BTA, a dopaminergic nucleus, we know that the chronic stress reduces the dopaminergic activity. And in the case for nucleus accumbens and hippocampus, we observe different anatomical effect of the chronic stress. For example, for hippocampus, we observe a decrease in the neurogenesis and a decrease in the dendritic tree. In the case for the nucleus accumbens, we observe that the chronic stress exposure produces an hypertrophy on the median spiding neurons. For these reasons, in this research, we assessed the activity of the prefrontal cortex and the BTA using the technique of symbolic unit recording, electrophysiological recording, and for the PFC, we record the local field potential. Given that this oscillatory activity has been associated with depressive symptoms. Also, given the anatomical changes in the nucleus accumbens and the hippocampus, we assess the activity of the AKT and GSK3 intracellular pathway and how CBD and THC formulation can modify these proteins and the neuronal activity of these areas. So the experimental approach is the following. After 1 week of habituation, the rats were exposed to different stressors, in this case during 2 weeks. Here we observe the stressor that we use, and after that, we carried out different behavioral tests to assess the acute effect of the CBD and the THC combination in anxiety and depressive-like behavior. After that, the rats were split in 2 groups. The first group was used to assess the expression and the phosphorylation of the AKT and GSK3 after an acute exposure of CBD. And the second group was used for electrophysiological experiment to determine the effect of CBD and THC in the activity of prefrontal cortex and BTA. Now, the results. Here, first, I'm going to show you the effect of the CBD and THC in anxiety-like behavior. Here, we observe the elevated plasmase. That is a test that the rat has to choose to explore a safe environment or an aversive environment. If the rat explores more time the open arms, that means that the rat shows a lower anxiety. For this reason, here we observe the effect of the different treatment in non-stressed rat, and here we can observe that with using all those, the CBD and the TAC and the combination doesn't show a significant effect of the open arm exploration. However, in a stressed rat, we can observe a significant increase in the open arm exploration after the acute exposure of CBD and the TAC combination. Now, another anxiety test is the social motivation. Here, anxiolytic drugs produce an increase of the sociability. In this case, the rat has to choose to explore a cage with a novelty rat or an empty cage. In this case, first, we can observe that the chronic stress protocol reduces the sociability of the rat. And then, when we use the treatment, only the TAC and the CBD combination produce a normalization of the sociability of the rat. Now, when re-exposed to the rat to the test, but using a familiar rat or an stranger rat, we can measure the social memory of the rat. And here we can observe that the drug doesn't change the social memory in the rat and in naive rat and also in a stressed rat. Now, to assess the depressive-like behavior, first we carried out the sucrose preference test. In this test, the rat, after 2 days of habituation to sucrose and overnight liquid deprivation, the rat has to choose to drink from a bottle of water or a sucrose solution. Normal rat prefer the sucrose solution. In this case, we can observe that the vehicle rat, in not stressed rat, prefer around the 80% the sucrose solution. And this preference was not modified by the drugs. And also, here we observe that the chronic stress exposure reduces the natural preference to the sucrose. However, different treatment, in this case, the CBD and TAC and the combination doesn't change the effect of the chronic exposure in the sucrose preference. Now, a second test to assess the depressive-like behavior is the forsuming test. The rat is exposed to a pool of water, and the rat can show mainly 2 strategies. The active strategy, in this case, climbing or swimming, or a passive strategy, the immobility. In this case, it has been, the evidence shows that antidepressant drugs reduce the time of the immobility, suggesting an association between the depressive-like behavior with the immobility. And here we can observe that the chronic stress protocol increase the immobility time in this test, suggesting a depressant effect of the protocol. Interesting, only the combination of CBD and TAC reduce the immobility time in this test. And taking together all of this behavioral evidence, we can suggest that we can observe a synergetic effect of the CBD and TAC in anxiolytic and antidepressant-like effect only with a pathological context, in this case, with chronic stress protocol. Now, showing the molecular effect of the stress and the CBD and TAC combination, we can observe here the expression and the phosphorylation of AKT in nucleus accumbens and hippocampus. Here we observe that the chronic exposure to stress increased the phosphorylation in both areas. And interesting, only in the nucleus accumbens, this phosphorylation was reversed by the drugs. Now, following with the GSK3, we observe a similar effect. The chronic stress exposure increased the phosphorylation of this protein in both areas, and now in both areas, the acute exposure to CBD normalize the phosphorylation of this protein. Showing again a specific effect of this drug in a pathological context. Now, when we carried out a single unit recording in the BTA of specific dopaminergic neurons, we observed that the chronic exposure doesn't change the basal activity of these neurons. However, the treatment increased significantly the tonic activity of the neurons in a stressed rat, showing again a specific effect of the treatment only in pathological context or on rat with a pathological background. Now, when we assess the activity of the pyramidal neuron from the prefrontal cortex, we observe that the tonic activity doesn't change with the chronic stress. However, the bursting activity is reduced after the chronic stress protocol, showing a decrease in the proportion of bursting cells and also in the spikes in burst. Interesting, with the treatment of CBD and THC combination, we can observe an increase of the firing rate in a stressed rat, and also a normalization of the electrophysiological phenotype in this area, increasing the population of population of bursting cells, and also increasing the burst activity in this area. And now, finally, when we assess the local fields potential in the prefrontal cortex, here we can see the power of different frequency. Here first, in the theta oscillation, we observe that chronic stress protocol reduce the theta power in the prefrontal cortex. We have to know that theta power has been associated with anxiety behavior. Also, interesting, with the exposure to the CBD and THC, we can observe a normalization of this oscillation. Now, when we see the effect of the chronic stress exposure in alpha power, this oscillation has been associated associated with depressive symptoms. The depressive patient has shown a decrease in alpha power in the cortex. And here we can observe a similar effects. The chronic stress exposure decreased the alpha oscillation in the prefrontal cortex. And however, in this case, the treatment of CBD and THC doesn't change this effect. And finally, with gamma oscillation, the gamma oscillation is very important given that antidepressant drug usually increase the gamma oscillation in the prefrontal cortex. And here we can observe the stress doesn't change this oscillation. However, the treatment of CBD and THC increase the gamma power in the prefrontal cortex, suggesting that the exposure to CBD and THC reorganize the neural circuit associated with the depressive behavior and anxiety behavior. Finally, the conclusions, CBD and THC edible formulation has a synergistic effect in anxiety and depressive-like behavior only in a chronic stressed rat. The formulation of CBD and THC reverse changes in the IKT, GSK3 pathway induced by chronic stress in the nucleus accumbens hippocampus. The combination increased the BTA dopaminergic activity only in chronic stressed rat, and the combination reversed the changes in the firing rate and increased the theta and gamma power in the prefrontal cortex. All of this evidence shows the therapeutic potential of this combination of drugs for pathologies associated with chronic stress exposure, for example, anxiety disorder or depressive disorder. Thank you so much. I want to thanks to my advisors, Steve Laviolet, Walter Roslo, and my lab mates, Hannah, Marta, and Roger. And finally, this research was funded by Canopy Health Innovation. Thank you.